Well, good afternoon, everyone. Thank you for joining us this afternoon to discuss the use of real-world evidence in HTA submissions. My name is Sam Gillard. I'm the Director of HTA here at Mtech Access, and I'm joined this afternoon by colleagues from both Mtech Access, Arcturis and Delta Hat, and we're delighted to sort of talk you through our webinar this afternoon. I'm going to briefly hand over to my colleagues to introduce themselves before we get started with the webinar. So I'm going to hand over to Dan first.
Thanks, Sam. Hi, I'm Dan Howard, Associate Director in Health Economics here at Mtech Access. I've got around 10 years of experience of technical modelling and consulting in industry, primarily for HTA submissions, including submissions to NICE, the SMC, and the NCP in Ireland. Most of the submissions I've worked on have been in oncology, and as a result, I have first-hand experience of everything that we'll be discussing today. I'll hand over to Joe.
Thanks, Dan. Good afternoon, everyone. My name is Joe, and I'm a Principal Medical Statistician at Arcturis. And in this role, I design and implement real-world evidence studies using the secondary electronic healthcare record data that we have access to at Arcturis. I'm looking forward to talking about both real-world evidence and real-world data today. I'll hand over to Nick now to introduce himself.
Thanks, Joe. Hi, everyone. I'm Nick Latimer. I'm a Professor of Health Economics at the University of Sheffield and also an Analyst at Delta Hat. So I'll be talking about decision-maker perspectives because I've sat on a NICE Appraisal Committee for 5 years. Back to you, Sam.
Thank you very much. I just wanted to say at this point, thank you to everyone who's already submitted questions ahead of the webinar. Please feel free, as we're going through the webinar, to add any additional questions to the chat. And as we get to the end of the webinar, hopefully we'll have some time to go through your questions. So what we're going to talk about today is I'm going to hand over to Dan to talk about partition survival modelling for HTA and some of the common challenges that are faced. Then we're going to talk about how real-world data can be used to generate external control arms. And as Nick said, he's going to be talking about sort of some real-world examples of how real-world evidence can sort of support economic modelling and some insight on how committees might see that. We'll loop back round for some reflections and next steps. And as I said, hopefully we'll have about 10 minutes at the end for some Q&A. So Dan, over to you.
Thanks, Sam. I should have control now. Great. So, in my section, I'll be doing a refresher on partition survival modelling. And more importantly, I'll be doing an overview of the types of challenges that we face and the evidence that we use to inform partition survival models, and in particular, how real-world evidence can contribute to these analyses. So, partition survival models, they're an ever more popular method of modelling cancer in HTAs. Most oncology appraisals for NICE and HAS use this approach. So the figure for NICE is 54% of 100 oncology STAs conducted between 2013 to 2018. STA is a single technology appraisal, utilize a partition survival approach. And this figure was 82% for in France for submissions to the HAS. That's between 2016 and 2022. They're used so often because they're conceptually really attractive. You only need 2 outcomes in principle, which are captured very frequently in cancer trials. And these are overall survival and progression-free survival, which is a composite endpoint of time to progression or death. With these 2 outcomes, you can model 3 health states. So you can see on the graph on the right, we've got progression-free, progressed disease, and death. However, you're rarely going to come across complete survival data where the Kaplan-Meier plots for OS and PFS reach 0 with no censoring. So in order to estimate what the full curves would look like, or rather to estimate them, we often fit survival models. There are lots of different methods for extrapolating these survival curves. That can be with independently fitted parametric models, jointly fitted models, some more complicated ones like flexible splines and mixture cure models, but they basically all attempt to do the same thing, which is estimate long-term outcomes from incomplete clinical data. These methods are all described in detail in the NICE technical support documents. If you want to go and have a look at them, which I recommend, that's 14, 21 and 24, which are all essential reading if you're going to submit to NICE and use a partition survival model structure. Once the curves have been extrapolated to the end of the time horizon that you're going to need to model, which is typically the entire lifetime of patients, you can categorize patients into health states. So here I've got a different graphical representation of a partition survival model, which illustrates the health states that we're going to model. These are the exact same ones that are in the graph on the right. And we've got the proportion of progression-free patients, which is just simply calculated as the area under the PFS curve. Then, to work out the proportion of patients who've died, that's calculated as 1 minus the OS curve, and that leaves us with the final health state, which is the number of patients who progressed but haven't died, and that's calculated as OS minus the PFS curve. So that very briefly has covered a simple partition survival model. You can have more complicated structure than this, but this is the most common structure that's typically used, the 3 health states. Right, now I'm going to cover some universal issues that you'll face in oncology modeling. So, as I mentioned, while the structure of these models can be quite conceptually simple, the main areas of complexity, and in my opinion, the really interesting part of this, is to do with the data that informs these models. So even a well-designed phase 3 trial can still have issues, like the wrong comparators being included, desirable comparators being excluded. For example, if you submit to NICE using an American trial, you might have comparators which aren't yet reimbursed in the UK. You can have heterogeneity in your trial population versus the target reimbursed population, and you'll definitely almost always have uncertainty around your long-term outcomes for OS and PFS. And this is an area where real-world evidence can really help fill these gaps. So, what to do if you have irrelevant comparators or a comparator that you need isn't included? So, as I mentioned previously, treatment pathways differ by market, and some treatments will be reimbursed in one market and not in another. So in these cases, you're going to lack head-to-head evidence. So you'll need to synthesize indirect evidence. Now, when it comes to indirect evidence, HTA bodies typically accept it, but historically this is focused on what I'm going to call traditional evidence synthesis methods. Things like BUCA indirect treatment comparisons and network meta-analyses, which typically require at least some direct evidence in order to build your evidence network with your comparators. However, this isn't always possible, and recently some other methods which do not require direct evidence have gained popularity, and these include things like matching adjusted indirect treatment comparisons, simulated treatment comparisons, and external control arms in particular, to name a few. These are all approaches where real-world evidence can be utilized and is really useful. You're also going to have uncertainty in your long-term survival curve extrapolations, which I mentioned earlier. This can be due to having immature trial data. You might also have strange behavior of your PFS and OS curves when they're estimated independently. For example, these curves can cross if they're estimated independently, so your PFS can exceed OS, which isn't possible. The subsequent treatments, the impacts of subsequent treatments, on OS can also have a factor in uncertainty in your long-term extrapolations, and uncertainty in the specific method that you're choosing. So the specific model that you're fitting to it is always a sticking point, or rather usually a sticking point in oncology appraisals. Just an example like a Weibull model, gamma, log normal, etc. However, real-world evidence datasets can address some of these concerns, often by a longer duration of follow-up and more complete survival data as a result. So another issue is treatment effect waning that we need to account for. This is particularly relevant in immuno-oncology, where trials often don't have a long enough follow-up to observe treatment waning, so we need to use assumptions. But we can inform and validate these assumptions by using real-world evidence and shed light on what treatment effect waning might look like in our population. And another area that I'm going to have a look at as well is, while I've been focusing on clinical outcomes, the other major components to a cost effectiveness model are obviously the costs. And historically, microcosting studies and key opinion leader views in ad boards, elicitation sessions, and interviews have been used to inform model costs, but real world evidence can potentially offer a solution here as well. So now I'm going to quickly go over some different types of evidence which are used in oncology modeling for HTA. These are going to be categorized broadly into direct and indirect evidence, which will lead on to the next section that Joe will present. So first of all, the evidence which companies are submitting for HTA is changing. Direct evidence is typically held as the gold standard with good reason, provided it contains the population and comparators that you're interested in, and in great enough quantities and quality. It's often straightforward to analyze head-to-head data as well, but this isn't always the case. And especially in recent years with legislative changes which have impacted the trial data which we end up submitting to HTA. So some of these changes include the innovative licensing and access pathway, which was introduced by the last government with the goal of speeding up marketing authorization. The current government have also cited earlier approval drugs as a goal in their life sciences strategy in the manifesto. In addition, we've got Project Orbis, which means that the FDA in the US and the MHRA can concurrently review new cancer drugs, meaning that rather than UK marketing authorization happening after US marketing authorization, this can potentially, if you go down the Orbis Type A route, happen at the same time. But why am I talking about regulatory pathways here, and why does this matter for HTA? Well, in the normal NICE STA pathway, the first committee meeting is normally held within a month of marketing authorization being granted. So there's a knock-on effect of having earlier regulatory approval with having earlier reimbursement and with earlier data, maybe quite a bit earlier than we've been used to using. Moving on. So, as I just said, HTA bodies generally prefer direct evidence for obvious reasons, but when you use earlier data, you're often using phase 2 data, for example, which might not have a comparator arm for technical or ethical reasons, and will often have lower patient numbers than phase 3 trials. So, in these circumstances, you'll need to rely on indirect evidence, which comprises of a variety of techniques which I've listed on this slide. It's not exhaustive, these are just a few that I've picked out. Even though direct evidence is often seen as more reliable, we can use statistical techniques, for example, matching and weighting, to decrease the impact of heterogeneity on our study results. So real-world evidence data sets have the potential to be a lot larger than anything you would see in a normal trial, and more complete because we potentially have a longer duration of follow-up as well, and maybe subject to less uncertainty when you're extrapolating long-term outcomes. NICE themselves have acknowledged the importance of real-world evidence and have published a framework to guide the use of it in appraisals. So where does real-world evidence actually fit in the context of what I've just described, in terms of the techniques we can use and the analyses that we can do? So here I've just got a very simple diagram of some indirect evidence synthesis methods. I've tried to categorize them into ability to adjust for heterogeneity across the top, and the data requirements for each of these methods in terms of cohort level versus patient level data down the side. So the top row here covers network meta-analyses and meta-regression, and naive comparisons in Bucher ITCs. Now, these are all extremely useful and well understood methods which make use of cohort level data, especially the network meta-analysis and meta-regression methods. But we can't always use these techniques due to a lack of head-to-head data, meaning that you can't form a network, or they might just be less desirable for whatever reason. For example, I would argue that a naive comparison is going to be less desirable than any of the other methods because you can't adjust for patient characteristics. These methods all have their place, however, they're just not the focus of today's webinar. Instead, we'll be focusing on these bottom ones which we've highlighted here. So matching adjusted indirect comparisons, simulated treatment comparisons, and in particular, and arguably the strongest method here, external control arms. These techniques are all data hungry, but they have huge potential for generating high quality evidence which can be used for HTA submissions. So now I'll hand over to Joe from Arcturis Data, who's going to talk about real-world evidence synthesis techniques in a bit more detail.
Yeah, thanks for that, Dan. That was a really nice overview of the current state of play. So as we've seen, real-world evidence may provide solutions to many issues that arise in the health technology appraisal process. And in this section of the webinar, I'd like to take a detailed look at how real-world data can be applied to achieve this. But what is real-world data and what does it actually look like in practice? Well, there are multiple ways in which real-world data is defined in literature, but most definitions do agree on the following aspects. So first, while real-world data can be obtained from several different sources, so things such as the electronic healthcare records or insurance claims data, a unifying feature is that the data has not been explicitly collected for a clinical study, but has instead been collected for purposes of things such as audit or reimbursement. Second, real-world data can take many forms. It may be structured data, such as quantified lab results or demographic information, or maybe unstructured free text, so things such as pathology reports or clinical notes. It's also the case that real-world data may be obtained from a single electronic healthcare record system, but it may also be constructed through linkage across multiple different systems. Third, real-world data will likely consist of individual patient-level data, but it may occasionally take the form of aggregate or summary data, although data in this aggregate format is usually going to have less utility than individual-level data. Fourth, collection of real-world data can be either retrospective or prospective, but it should always capture the realities of routine clinical care. And finally, due to the way that real-world data is collected, any analysis that makes use of this type of data will have to be observational in nature. But I think it's fair to say that the most common and arguably the most useful real-world data sources will be derived from retrospectively collected electronic healthcare records and will consist of individual patient-level data. So as the quantity and quality of information stored within electronic healthcare record systems grows, the breadth of insight that could be obtained from this data source will grow too. The information content of this data is now so great that it often allows us to perform indirect treatment comparisons with clinical study data. And this is particularly useful, as you've heard, in situations where a randomized clinical study may not be possible for ethical or logistical reasons. And an increasingly popular approach to performing this type of indirect treatment comparison is through the use of something called an external control arm, or ECA. So what is an ECA? Well, as with the concept of real-world data, there are a few ECA definitions that are present in the literature, but broadly speaking, an ECA is a control group consisting of patients who have been sourced from outside of some clinical study of interest who have individual level data available. Now, this individual level data will have been used to assess each patient for the eligibility criteria of that clinical study of interest. The patients who fulfill these criteria will then constitute that external control arm. They can act as a control for the clinical study of interest. And this is achieved through a comparison of outcomes between the external control and the patients who have received an investigational therapy in the clinical study of interest. I think to understand the role played by an ECA, it's important to first consider the gold standard of clinical evidence generation, which is the randomized trial. In a randomized trial, a control and intervention arm are constructed from patients who fulfill the study eligibility criteria, with assignment to each study arm being performed at random. This ensures that the received treatment depends purely on that random assignment, allowing us to assign a causal interpretation to any differences in outcomes that we observe between these two study arms. Now, as you've seen, unfortunately, a randomized study is not always possible. This can be due to ethical or logistical constraints. And in such cases, a single arm trial may instead be used to demonstrate the safety and response of a novel therapy. In a single arm trial, again, a cohort of patients are selected based on their fulfillment of some eligibility criteria, but all patients in the study will receive that investigational therapy. There's been recent interest in safely expediting the health technology appraisal process using these results from single arm trials. This is achieved by taking the single arm trial data and performing that indirect treatment comparison with some external control, with the ECA a clear option for this process. Now, in the real world, patients will receive therapy based on clinical decision-making and reimbursement guidelines. But if the eligibility criteria of the single-arm trial are applied to these real-world patients, we can effectively filter them down to create an external control arm consisting exclusively of patients who could have hypothetically participated in that same single-arm study, but who have actually received usual care instead of a novel treatment. If data from this ECA is then combined with the single-arm trial, The use of statistical causal inference methods can allow for a comparison of outcomes in which the bias introduced by baseline differences between the cohorts can be mitigated, allowing us to generate insightful indirect treatment comparison evidence. So ECA analyses are a particularly impactful application of real-world data in the HTA process. And this is because that indirect treatment comparison that's being conducted with an external control arm can really integrate large quantities of representative data from real-world sources, providing estimates of the effects of a novel treatment relative to one or more therapies currently being used in routine care. But how can we actually construct an ECA analysis in practice? And what should guide our decision-making when applying concepts we see in randomized studies, such as eligibility assessment, to real-world data sources? Well, the document that can help guide us in this process is NICE's real-world evidence framework. This describes best practices for generating high-quality evidence from real-world data. Obviously, it covers a range of topics, but a fundamental aspect of a real-world evidence framework is the use of something called the target trial emulation framework. This is particularly relevant when conducting an indirect treatment comparison. So the target trial emulation framework explains that analyses utilizing an external control should aim to actually emulate the randomized study that ideally would have been performed instead of a non-randomized study that was actually performed. So to achieve this, we must consider all aspects of the construction of an external control arm and do our best to align them with that hypothetical randomized study we would like to have performed. Of particular interest here are the ways in which we should perform eligibility assessment, how we should define the study index date, how study outcomes can be harmonized across real-world and trial data sources, and what our choice of statistical method will be to perform that comparison of clinical outcomes. So, to demonstrate best practice for ECA construction, I'd like to focus on a fairly recently published study that incorporated many aspects of a target trial emulation framework into its design. So in this study, the authors compared participants who participated in ZUMA-5, which was a single-arm study of axi-cel for follicular lymphoma, with an external control arm consisting of patients sourced from electronic healthcare records who had received routinely used therapy for follicular lymphoma. The authors called their ECA cohort the SCHOLAR-5 cohort. They compared multiple categorical and time-to-event outcomes between the ZUMA-5 and SCHOLAR-5 cohorts. To handle differences in these cohorts at baseline, the authors used propensity score-based methods when comparing outcomes, and this allowed them to adjust for differences in the demographic and clinical profile of patients across these two study arms. Now, as mentioned previously, the design of an external control arm can be deconstructed into several concepts, each of which align with the different aspects of a target trial emulation framework. And what I'll do on the following slides is detail how this study was conducted and how the study design aligned with those key concepts from a target trial emulation framework. So for this ECA analysis, the first concept to consider is the assessment of each available real-world patient for the eligibility criteria of ZUMA-5. Now, due to the prevalence of missingness in electronic healthcare records, it's not always possible to assess all eligibility criteria when using real-world data. So it's important to identify which criteria can be assessed directly, which cannot be assessed, and which criteria may be assessed using proxy criteria that are constructed specifically to work with the content of real-world data sources. When generating the SCHOLAR-5 cohort, the authors were able to directly assess many of the ZUMA-5 eligibility criteria directly, including things such as the number of prior therapy lines each patient had received. They couldn't assess criteria such as whether patients were pregnant or breastfeeding. This is because this information may not always be easily determined from electronic healthcare records. The authors also built a proxy assessment criteria for ECOG performance status being 0 or 1, and they did this by converting Karnofsky scores, which is a similar measure to ECOG, into ECOG scores where ECOG scores were not available, but Karnofsky scores were, and then assessing these new observations for the eligibility criteria. This approach of using proxy criteria where necessary makes best use of all available data and allows for a more thorough assessment of eligibility. Following the construction of the ECA eligibility criteria, it's necessary to identify when the follow-up period for each real-world patient should start, and usually this index date will coincide with the initiation of some comparative therapy of interest. In SCHOLAR-5, the comparator was any therapy that was routinely used for the treatment of follicular lymphoma that was initiated after eligibility was first achieved by a patient. In this analysis, that meant that multiple index dates were actually possible for any patient who had received more than 3 prior lines of therapy, as any subsequent line could actually be used as a comparator. So the presence of multiple potential index dates is a common issue encountered when constructing an external control arm and can lead to the introduction of immortal time bias if not properly accounted for. In this study, the authors randomly selected the start of one eligible therapy line as the index date for each patient, and this represents one of a number of approaches that are applicable in the target trial emulation framework that can be used to circumvent the issue of multiple index dates. Once the index date can be identified for each real-world patient, the next step is to capture key eligibility and measured confounder data around that index date. So to do this, a capture window is defined around index date, and data points that reflect the status of the patient at baseline are retained. This diagram, we can see how ECOG and Karnofsky score data may be captured around index date, enabling assessment of whether each real-world patient had an ECOG score of less than 2 at index date, and consequently whether that patient fulfilled this eligibility criteria for entry into the external control arm. Now, importantly, multiple relevant data observations may be made during the capture window, and rules must be defined during the study design stage to ensure that eligibility is assessed using only the data that's most reflective of the status of each patient at index date. In the example presented on this slide, we can see multiple observations captured around index date, but only those that are closest to index date, and preferentially those before index date, are used to assess that eligibility. Once eligibility and confounding data have been captured, it's possible to apply the eligibility criteria themselves and actually identify those patients who will form that ECA. Once the ECA cohort is identified, the next step is to construct outcome measures for these patients. In the Zuma Scholar 5 study, the authors considered standard clinical trial outcome measures, including progression-free survival and overall survival, alongside therapy-based proxies of clinical trial outcomes. And in this case, the authors considered time to next treatment, which often acts as a proxy for progression-free survival, as progression is usually closely followed by a change in therapy. So that change in therapy is then taken to be indicative of a progression event having occurred. On this diagram, we can see that in certain scenarios, there may be a considerable time difference between observed disease progression and any change in therapy that may be used to construct a proxy outcome, such as time to next treatment. And because of this, where possible, the outcome measures that are compared between trial and real-world sources should be constructed using the same criteria. This approach is preferable as it allows for a like-for-like comparison between trial participants and ECA members. And this reduces the possibility of systematically inflating event times in the control arm, which could bias any estimated treatment effect. And at this stage, it's also important to determine how censoring will be handled for the external control arm cohort. For most electronic healthcare record-based studies, the censoring date will reflect the data which data has been cut by the data provider and received for analysis. But it's also important to consider whether patients have become unobservable for any reason before that data cuts. And then the final step in ECA analysis is actually that comparison of outcomes between the external control arm and the trial cohorts. Importantly, in this case study, the authors did identify a series of confounding covariates that included prior stem cell transplantation, relapsed or refractory status at baseline, and the number of prior therapy lines, amongst others. These covariates have an association with both the treatment assignment process and the measured outcomes, meaning that they were confounders and that they needed to be controlled for to minimize bias in any estimated treatment effect. By capturing these covariates around index date, applying them in a model which produced estimates of propensity scores, and then using these scores to reweight the contribution of each patient to the analysis, the influence of measured confounders in comparisons of outcomes was mitigated in this case study. Now, in any ECA study, it's imperative that the choice of confounders included in that analysis are based on clinical expertise and careful consideration of how all measured variables are associated with the treatment, the outcome measure, but also with one another. And in this case, after following key aspects of the target trial emulation framework, the authors did identify a clear treatment benefit for ICER versus usual care for follicular lymphoma, with a progression-free survival hazard ratio estimated between 0.18 and 0.49. And as you can see on this Kaplan-Meier plot, there's clear separation between these curves. So we've seen how an ECA can be constructed from real-world data and an overview of what constitutes best practice for the design of an ECA study for an indirect treatment comparison. But it's important to recognize that real-world data can provide important evidence across the wider HTA process too. So for example, construction of a cohort of patients receiving a comparator therapy for a given technology appraisal enables clear characterization of real-world comparator use, and this includes assessment of the use of support medications, healthcare resource utilization, and capture of the wider costs associated with that comparator. Similarly, real-world data can be used to generate evidence to support specific economic modeling assumptions regarding comparator use that may have a meaningful influence on the ICER. Now, so far, we've seen some potential uses of real-world data in the HTA process and how this data should be applied, but fundamental to the use of this evidence is establishment of the suitability of the data and statistical methods that have been used to generate it. Ultimately, regulatory and reimbursement decision makers must make this assessment, and there are many considerations they will make when determining whether the evidence they've been presented with is of suitable quality. So I'll now hand over to Nick from Delta Hat, who will talk us through what's involved in that assessment process.
Great, thanks very much, Joe. I think I've got control now. Yeah, okay, great. So, as I said before, I'm a professor of health economics. Oh, sorry, I'll turn my camera on. I'm a professor of health economics at the University of Sheffield and also an analyst at Delta Hat. And I'm going to be giving a reviewer or decision maker's perspective on the use of real-world data, real-world evidence to support economic modelling for HTA. So just before I start, some quick disclosures. I was a member of one of NICE's appraisal committees for 5 years. I'm a member of NICE's decision support unit. Sometimes I work with Sheffield's technology assessment group, and in fact Delta Hat acts as a NICE external assessment group in collaboration with Pentag at the University of Exeter. Also, I'm focusing my slides on the NICE real-world evidence framework, which has already been mentioned today, and my experience on NICE committees. But what I present, what I say, just reflects my own opinions, not those of NICE or anyone else. Just want to make that clear. Okay, so when we're considering the acceptance of real-world data analyses by health technology assessment agencies, I think the NICE real-world evidence framework is a crucial document. Obviously, it is specific to NICE, but for many of us, NICE is a key agency. And I think a lot of what the document says is also representative of the views of several other HTA agencies around the world, although, of course, not all agencies will agree on everything. In my view, the real-world evidence framework document is a bit like the technical support documents produced by NICE's decision support unit. And I think it's a document that review groups and committee members will refer to for guidance, and so will inform decision making. So the real world evidence framework has a key messages section right at the start, and that really highlights NICE's commitment to using real world data within technology appraisals. So it says, as described in the NICE strategy of 2021 to 26, we want to use real world data to resolve gaps in knowledge and drive forward access to innovations for patients. We developed the real world evidence framework to help deliver on this ambition. The framework document then goes on to explain how it will deliver upon that ambition by identifying when real-world data can be used to reduce uncertainties and improve guidance, and by clearly describing best practices for planning, conducting, and reporting real-world evidence studies to improve the quality and transparency of evidence. And it then says that the key aim of the document is to improve the quality of real-world evidence informing our guidance. So the framework document then goes on to describe 3 core principles around the use of real-world data to inform appraisals, and I think these are really important. So firstly, we must ensure data is of good provenance, relevant, and of sufficient quality to answer the research question. Secondly, we must generate evidence in a transparent way and with integrity, right from study planning through to study conduct and reporting. And thirdly, we must use analytical methods that minimize the risk of bias and characterize uncertainty. So I think it's obvious that if a company wishes to use real-world data in an appraisal, NICE's review groups and appraisal committees are going to want to see evidence that each of these core principles has been adequately covered. So to think about what that means in a bit more detail, I'm going to go through each of these principles a little bit more. Okay, so firstly, data provenance, relevance, and quality. To address this core principle, the framework document helpfully includes DataSat, which is a data suitability assessment tool which covers data provenance, quality, and relevance. So for data provenance, information must be provided on the data source and on things like data management and governance. For data quality, information has to be provided on the completeness and accuracy of each key study variable, which are those variables that are particularly important for the analysis, so such as variables on interventions received, outcomes, and prognostic characteristics. For data relevance, the population and setting of the data needs to be described so that a reviewer can see if the data are really relevant for the decision problem being addressed. So, for example, this could be about whether the real-world data source is from the NHS or from a population that is similar to the NHS. Now, I think it's important to highlight that completing datasat isn't a requirement when using real-world data to inform a NICE submission, but it certainly makes sense to complete it if you want to increase the chance that NICE will deem the evidence reliable enough to inform decision making. If NICE provide the tool, it makes sense that you should probably use it. Okay, so a bit about my experience with key issues for reviewers and decision makers on this particular topic. Firstly, I think committees almost always think about where the data are from and whether the data are generalizable to the population of interest, which is really about the relevance of data. So, for example, NICE is making decisions for the NHS population, so wants to see if the real-world data set is generalizable to the NHS population. If the real-world data source is from the UK, then people in it are likely to be selected from that NHS population. But then the question is whether all types of patients in the NHS population are represented, or does the real-world data have specific eligibility criteria, which could mean that there are important prognostic differences between the patients in the NHS population and the patients in the real-world data source. So NICE are always going to think about this. Or if the real-world data is from another country, committees will think about whether the data are transportable to the NHS population. And transportability is another thing that has been added quite recently to the real-world evidence framework document. So again, they'll think about where is that data source from, and are the populations similar to the target NHS population, or do they have important differences? Beyond where the data are from, I think it's very common for NICE committees to consider the variables included in a real-world data set. So are important variables measured? And amongst the variables that are measured, is there any important missing data, which is about the quality of the data? And fundamentally, the reason committees care about missing data and the variables that are measured is because if they think that there could be important differences between the population in the real-world data set and the NHS population, or in the case of external control arm studies, the population in the uncontrolled study of the experimental drug and the population in the external control arm, then the committee needs to know whether the data are sufficient to allow adjustments to be made to account for the differences in study populations. So what this fundamentally boils down to is that the committee, and this is my chair of committee here, are going to want to know whether they can really rely on the real-world data sources and whether the data are relevant enough to address the decision problem being faced in the appraisal. And it's likely that the committee will probably think that if the data aren't relevant or are of poor quality, that they can't rely on it for decision making. So I've dwelt on this data provenance, relevance and quality aspect here quite a lot because I think it's really important. Sometimes it is tempting to spend a lot of time thinking about exciting, intricate analytical methods, which are important. But if the data aren't right, analyses won't be useful no matter what we do. And I think that is important to bear in mind. Okay, so the next core principle covered by NICE's real-world evidence framework is study planning, conduct and reporting. So obviously the framework document says a lot more than what I have on the slide here, but a really key part is that as much as possible should be pre-specified and published, and that includes the study design, analysis plans and reporting. The document recommends the Harper tool for developing protocols for real-world data studies and refers to several reporting checklists as well to ensure that all important aspects are reported clearly and transparently. Well, I think most of this will be naturally covered if a target trial approach is used, as explained by Joe. But still, it's important that all of this is planned up front and that it's documented transparently. Now, I think the key questions that committees often ask around this is whether analyses were pre-specified, based on a concern that if they weren't, perhaps they've been cherry-picked to provide the desired results. And related to this, the committee will want to know that the study has been designed and conducted in a way that's going to minimise bias. And for the committee to be confident about that, the study needs to be planned, conducted and reported really clearly. Okay, so the final core principle covered by the NICE RWE framework about analytical methods. So Joe has already talked about the methods, so I'm not going to go into detail here. But some key points are that the framework recommends taking a target trial approach, using analytical methods that avoid or remove bias, and including sensitivity and scenario analyses to give an idea of the stability of the results and the importance of any potential biases that might remain due to things like missing data or unmeasured confounding. I think that depending on the expertise of members of the review groups and appraisal committees, there are a whole range of questions that could be asked about the analytical methods used, around things like choices of time zero time points, variable selection, missing data, treatment pathways, censoring, potential residual biases. So lots could come up there. Okay, so before I finish, in Joe's slides, he talked a little bit about a published external control arm study for axicabtagene ciloleucel for follicular lymphoma. I thought it might be interesting to look at the NICE appraisal that used that external control study to see what kinds of things the NICE Appraisal Committee discussed in that case. So this was Technology Appraisal 894. You can look it up. And it's important to say that my summary here isn't exhaustive. These are just some bits that came directly out of the final guidance document on the website that I thought were interesting. So first of all, about relevance of the real world data. If you remember, AxiCell had been investigated in an uncontrolled study called ZUMA-5, and an external control arm was created using real world data in a study called SCHOLAR-5. The NICE committee concluded that using data from SCHOLAR-5 was acceptable, although it did notice a mismatch between the standard treatments received in SCHOLAR-5 and in the NHS. So there were some differences. With reference to the conduct of the ECA study, the committee discussed the eligibility criteria used for people included in the comparative effectiveness analysis, which was important because the number of prior treatments did differ in the two studies. The guidance document also noted, and I thought this was interesting, the methods used to adjust for baseline differences between the ZUMA-5 and SCHOLAR-5 populations, and that these methods had improved the comparability of the data sources. But there was also quite a detailed and pragmatic, I think, discussion about covariate selection, where it was noted that adjusting for lots of variables is difficult when sample sizes are small, as they were in some cases here, because this can lead to problems with convergence of the statistical models. So the committee was clearly thinking quite hard about this and in quite a lot of detail. With respect to the reporting and I suppose conduct as well, the committee noted that the company had submitted sensitivity analysis using alternative methods and quantitative bias analysis, both of which were seen as good things. Overall, the committee's conclusion on the analyses, at least the conclusion recorded in the guidance document, is, I suppose, a little bit vague. It stated that the adjustment method used by the company is highly complex. It's not really directly saying whether the analyses were acceptable or not. But it is clear from the document that the analysis was used for the decision making, although the lack of direct comparative efficacy data was highlighted as contributing to a high level of uncertainty in the appraisal, which I guess is what you would expect. Okay, so finally some conclusions from me. So although this session is about using real world data in HTA, I think it is important to remember that RCTs remain the preferred source of evidence on the effects of new treatments, and that is stated in NICE's real world evidence framework document. However, it is also stated that non-randomized evidence can be used when RCTs don't exist or aren't relevant, or are of poor quality. So certainly we can use these data sources and analyses. HTA decision makers are generally open to this, but we can't just use any old RWE study. The data needs to be high quality and relevant, and the analyses need to be well planned, pre-specified, and well reported. And finally, even though I am a fan of using complex methods, it's what I've spent a lot of my career trying to do, a lot of my research is on these methods and these data sources, we do need to understand that when we're not using randomized data, even high quality analyses using high quality data are likely to be associated with high levels of uncertainty. And I think it's fair to say that decision makers may be more aware of uncertainty and perhaps will be more tentative when making decisions based on real world evidence rather than RCT evidence. And with that, thanks very much for listening. And I think we can move on to questions.
Thank you very much, Nick, and all the speakers. That's been absolutely fabulous. So yeah, I'm going to open the floor to some questions, and I'm going to start with some of the, well, actually, I'm going to have a little bit of a reflection before we add on questions. So I think today, and thank you again to all our speakers, we've learned a bit about using real-world evidence in oncology. We've learned about how external control arms can be devised, sort of the best methods for that, and then some real-life examples of that. So thank you very much. And then some real critique of how an EAG and a NICE committee have critiqued some of that real-world evidence. So that's perfect. Thank you very much. So I'm going to start with some questions now. And I think the first question to you, Dan, can you explain how some of the concepts that we've covered can be applied in non-oncology settings? So, for example, rare disease or nephrology?
Yeah, sure. I think that it's important to state that we picked oncology here because it's a really good example of a disease area where you're going to have a lot of data sources available and that the outcomes that you need are very well characterized and often easily attainable. I say that sometimes PFS isn't the easiest to get and sometimes you have to use proxies for it, like time to discontinuation, but there's no reason why any of these principles that we've been describing today can't extend to other disease areas. I think the main issues that you're going to run into are the evidence sources themselves. So in rare disease, for example, you're probably, by the virtue of it being a rare disease, you're probably going to find less real-world evidence than you would in a more common disease. And it really depends on the registries that are available and the outcomes as well that you're measuring. So that's why we picked oncology, because OS is an objective outcome. It's easy to assess, you know, a patient's alive or they're not. And a lot of the other outcomes that you find in oncology real-world evidence sources are very well characterized, understood, and have a large amount of data available.
Thanks, Dan. And then question for you, Joe. So sort of building on this, we also asked about the use of real-world evidence in the case of missing comparator data in hard-to-research areas. So any sort of thoughts on that?
Yeah, I guess my answer is going to be quite similar to Dan's one because the questions kind of overlap a bit. Maybe I can give a perspective from somebody who has their hands dirty with the data. So yeah, obviously the example that I used wasn't in a rare disease or anything like that, but it was in an oncology setting. And I think, as Dan said, it would be fair to say the majority of examples of real-world evidence use are in the oncology setting. And this is a consequence of the fact that a lot of the data that is generated in the oncology setting is required to be collected for audit purposes, so it's there, it's available. But also a lot of the single-arm trials are also in the oncology setting as well. But having said that, obviously there's no reason the approaches that have been discussed today couldn't be applied to rare diseases, and we do see it happening in practice. But I think you have to accept you're going to see small patient numbers, and therefore you're going to have high variance in the evidence that's been generated. But insightful evidence regarding comparative use, best supportive care, etc., is definitely achievable, particularly with larger data sets that can capture many patients. But they have to have that sufficient granularity that you can assess some of those more complex clinical outcomes that are often associated with rare diseases. At Arcturis, we spend a lot of our time actually collecting and cleaning that type of data that's required to construct those types of outcomes. That data is out there, it just requires a bit of work to get it. And then from like the statistical perspective as well, rare diseases offer some slightly different approaches. So we might be inclined to reach for some slightly different methods. So things such as Bayesian approaches, they're particularly appealing due to their ability to incorporate prior knowledge from other studies in a principled way. This should allow us to minimize some of the uncertainty associated with the small numbers and really squeeze as much information as possible out of the data that's there. But yeah, in general, rare diseases, harder to research areas are complicated by small patient numbers, requirement of often quite complex data and occasionally complex statistical methods. And in my experience, earlier the process for real-world evidence generation sort of integrated into the development of the HTA submission in a rarer disease, and the higher the quality of evidence that's going to be produced, because more time can be taken to actually identify the right sources and the right statistical methods to generate that evidence.
Thanks, Joe. So Nick, we've had a lot of questions about how HTA agencies might view real-world evidence. And what do you feel are the sort of unmet opportunities for HTA agencies when it comes to real-world evidence?
Yeah, okay. So, I mean, I think the real-world evidence framework document demonstrates that these agencies are thinking a lot about these data, and it's not uncommon for these data to be used. So, use is being made. My personal opinion is I think there's two areas where they could use it a lot more. So the first is in terms of reviews of previous decisions. At the moment, a lot of NICE decision making, NICE recommendations say they'll be reviewed in X number of years, 2 or 3 years or something. But quite often that's not really done in a particularly meaningful way. And I think it could be using real world data. Once, if a treatment had a positive recommendation, then it's being used for that number of years. You can look to see what's actually happening and review the decision. I think that would be useful. And then the more kind of obvious one is the Cancer Drugs Fund appraisals, where the drugs in the Cancer Drugs Fund for a while, normally 2 years, sometimes longer. And then when the review is done, one of the key data sources is from SACT. But what you find is that the data from SACT is always just for the experimental drug, which leaves you with problems in terms of if you want to use that data to see what's the treatment effect in the real world, you can't really just with that data. So obviously the data on the comparator is there from prior years before that new drug had been kind of put in the Cancer Drugs Fund. I would like to see much more use of comparative effectiveness analyses using data from both the comparator and the experimental drug.
Yeah, and as you say, that data should be there, so why not use it? Yeah. Okay, question for you, Dan. Have you had a sort of direct experience with NICE accepting real-world evidence that changed, the committee's decision making?
Yeah, well, I think we've seen an example that Nick just went through of an appraisal where real-world evidence was used as part of the main evidence submission. And speaking from my own experience, I think I've taken part in NICE submissions that I don't think would have been possible at all without the use of real-world evidence. I simply don't think that it would have gone through. You could have made an evidence submission because there would have been no comparative evidence available. So I think more than just influencing a decision, I think it also makes it possible to make a decision as well. Or rather than the appraisal not going ahead at all, I think it may have happened just much later in the future when you have a phase 3 trial. And all that time, there are patients who are missing out potentially on access to drugs when potentially access could have been achieved earlier with the use of real-world evidence. Yeah, it definitely does influence the decision making, and it even makes it possible to make a decision.
Thank you. Joe, question for you, sort of looking beyond HTA, how can real-world evidence contribute to sort of long-term monitoring of health technologies post-approval, and what role can it play in reassessing technologies as new health data emerges?
Yeah, so I mean, naturally, real-world evidence does play a key role in post-authorization studies, and I think it'd be fair to say this is probably the most established use of real-world data in a regulatory setting or a reimbursement setting. And as information content in the electronic healthcare records is only growing, sort of the effectiveness of these types of analysis will also only grow. But I think in general, I would actually just agree with a lot of what Nick was saying in his answer previously. The Cancer Drugs Fund is a really clear example of post-authorization use, really, where the drug has entered through a patient access scheme and data is being collected in a real-world setting, and then a number of years later, that data is being reassessed. And I would completely agree with what Nick said about the comparator. That data is sat there. The same data that's being collected for the novel therapy is being collected just through routine care for the comparator. And this same principle applies essentially for any situation where there's been a recommendation from NICE and the drug has entered routine commissioning. All of the drugs that are involved in that process that are being used in the NHS will be generating data in the electronic healthcare records, and that data is available for reassessment at a future date if required. So yeah, I'd be completely on board with Nick's answer to his question, and the similarities to the question there about post-authorization. Yeah.
Okay, great, thank you. I think we've got time for a couple more questions. I guess coming back to you again, Joe, sorry to pick on you again, but what role can machine learning or AI play when we're generating real-world evidence to support a HTA process?
Yeah, I mean, so obviously like AI and ML are like hot topics at the moment, and it seems like these concepts are being applied everywhere. But I think when it comes to the assessment of novel therapies, we do need to be careful about where these methods are applied. Many of these methods are essentially like black box methods, meaning you don't really have any understanding of how they've arrived at a result that they've generated. And when it comes to assessing whether a technology is effective or safe, it's not particularly reassuring. So yeah, the more traditional statistical methods, parametric approaches, survival curves, and so on, they're here to stay because they have clear assumptions about how the data is being modelled, and they can be tested directly. So you can assess whether these methods are appropriate for their use. But having said that, I think there are some areas where AI or ML methods do have some utility. And I think a really clear example is data enrichment and content extraction. So at Arcturis, we have an ML team who work with the free text documents that we access. We've got sometimes tens of thousands of these, and we need to parse them quickly, and they can develop these ML models which can extract the key data from these unstructured sources, turning them into structured sources that can then be leveraged for analysis. Now, obviously, there needs to be some assessment of the precision and accuracy of these methods, but that's completely something that can be done. So I think our ability to take an electronic healthcare record system and turn it into an analyzable data source using AI methods is a potentially really, really useful application.
And then following on from that a little bit, I know NICE have just sort of released a bit of a statement on the use of AI, but it's a little bit unclear and a bit woolly. But do you think that will be something that in the future NICE will be more happy to accept? And I guess I'm opening the floor to any of our experts on that one. Don't know, Nick, if you've got a thought on that.
Yeah, I mean, it's not specific to AI, but I think it's also relevant to AI and any methods that use things like that are quite novel. Like a lot is down to reporting, I think. Quite often when complex methods or new methods are used in submissions, they're not actually that well reported. And so for a reviewer or a committee member, it's very difficult to know to what extent you can trust these analyses if they haven't been reported well. So I think that's a really key thing. The other thing is proof of concept that these methods work is an important thing. So more broadly than just the AI stuff, but benchmarking studies are things that people have started doing more. So in the US, there was something called the RCT duplicate study, where they used US health data to try to emulate existing RCTs and see if you could get similar results using the real world data. We're doing similar work to that using English cancer data at Sheffield, actually. But I think if you want to use a data set, proving that it can give reliable results by benchmarking against an existing RCT and then doing the analysis you actually need to contribute to the submission you need it for is also a good process, which I don't think has been done much.
Okay, well, we're just up on the hour, so I just want to wrap up by saying thank you, everybody, for attending the webinar. The webinar is available, I think, on one of the links at the top in the materials, but we will be following up with everybody. And for everyone who has put in questions that we haven't got round to answering, we will endeavour to follow up with as many questions as possible within the next 2 weeks. So thank you very much for your time this afternoon.